Since I will speak about neutrophils, I would like to give a brief introduction about its antimicrobial functions. So neutrophils clear bacterial infection by the following ways. It produces granules. You can see pinkish and bluish colored things inside the neutrophil. I would like to give a brief. These are the granules. It produces granules containing antimicrobial peptides, which when released can kill bacteria. That is number one strategy. It can also engulf bacteria via phagocytosis and kill the bacteria. That is the second strategy. And recently, scientists found another phenomenon called netosis, which is a third strategy where neutrophil release its genomic DNA, which will entrap the bacteria like a spider web entrapping insects and kill them. So, like I told earlier, the role of neutrophils in TB is not very clear. And the very best way to study its function is through direct visualization of its behavior. Next slide, sir. So currently, mouse model is the widely used model to study TB infection, but direct visualization of neutrophils is not possible in this model, mouse model, because it is opaque. Also, mouse model of TB infection does not produce granuloma, the hallmark structure of TB, which I introduced in my previous slide. So, in this study, we use a new and unique zebrafish and Mycobacterium marinum infection model to study TB-associated neutrophil functions for the following reason. So, the Mycobacterium marinum is a natural pathogen of fish. Its genome is very similar to Mycobacterium tuberculosis, the causative agent of tuberculosis. So that means that you can instead use Mycobacterium marinum to study function of MTB because they are very close. And second thing is it forms Mycobacterium marinum when it infects zebrafish, it forms granuloma which is similar to human granuloma. So you can see on the left side, adult zebrafish granuloma stained with H&E staining. And on the right side, you see a human granuloma, and you can appreciate that both have a very, very similar structure with a central necrotic core surrounded by immune cells. So this means there is a very good model to study TB. So now the question is, how can we visualize neutrophils in this model? Next slide, sir. So for this, you have to first specifically label the neutrophils. So how can you do that? So you need to put a fluorescent gene under the control of a neutrophil specific promoter, which should be active only in the neutrophil. And when you inject these DNA construct into zebrafish embryo and make further genetic crosses, you will get zebrafish with fluorescently labeled neutrophils, like you see at the below in the slide. So the next problem is that you need to find a way to take out the granuloma from the infected fish, maintain them alive for a very long time to visualize neutrophils in them. So how will you do that? Next slide, sir. So for this, we devised a strategy where you infect adult zebrafish with Mycobacterium marinum, wait for 14 days for them to form granuloma, and then euthanize them, that is kill them, dissect the granuloma and take out the granuloma, and maintain the granuloma in what we call as a 2.5D culture media, which is a media enriched with extracellular matrix like collagen. So these granulomas can be kept alive in this media for 5 days and can be imaged using fluorescent microscopy. We call this model mycogem for mycobacterial granuloma explant model. So now we have the model ready. The next step is to visualize fluorescent neutrophils in this model. Next slide, sir. So on the right side, you see a granuloma with neutrophils labeled green and bacteria in red. So on the left side, it is the same granuloma, but I removed the bright field image so that to just show neutrophils and bacteria. So now sir is going to play a movie where you will see dynamic behavior of neutrophil in the granuloma. Sir, could you please click the left image? Yeah, you see the play button. Yeah, just thanks, sir. Right. So this is the first time anyone has seen live action of neutrophils in the granuloma, and you can appreciate from this movie the dynamic nature of neutrophils, which is contrary to the old belief that they are very static in nature. And the next question is, what are the physiological roles of neutrophils in the granuloma? Next slide, sir. So to answer this, we examined more granulomas, and here you are seeing one such granuloma where you could see the central necrotic core with a lot of bacteria as indicated by yellow arrow. You could also see a lot of bacteria present outside the necrotic region indicated by white arrow, which are pathological hotspots, and if left uncontained, these bacteria will grow and expand and worsen the infection. Now sir is going to play a movie, and in this movie that I'm going to show you, you will see that neutrophils go and lies near the region containing bacilli outside the necrotic core indicated by white arrow. Sir, could you please play the movie? This slide, same slide. So you can see that the neutrophils go and lies near the region containing bacilli outside the necrotic core indicated by white arrow. So you can also watch the movie again if you want to be clear. Yeah, now you can clearly see the neutrophils lysing near the bacteria. A lot of neutrophils actually lysing. So what does it mean? It means that when neutrophils lyse in this region, it releases antimicrobial proteins from granules that I mentioned earlier, and these proteins will inhibit bacterial growth, controlling the infection. So by this action, neutrophil protects the host. Next slide, sir. So these are just still images from the movie that I showed you before. And here you can clearly see that over a period of time, which is noted at the top of each picture, neutrophils undergo lysis near the bacteria as shown by white and red arrows. So next, if you remember my earlier slide, granulomas are nothing but organized aggregates of immune cells, and hence it is possible that neutrophils could interact with other immune cells in the granuloma, and these interactions could further affect the outcome of infection. So we looked for interactions between neutrophils and other immune cells, especially macrophages, which are present in majority in the granuloma. Next slide, sir. So here on the left side, you could see a granuloma with neutrophils labeled in green like before. In addition, you could see macrophages labeled in red and bacteria in cyan. So images in the right panel B shows magnified version of the boxed region shown in the left over a period of time. So here you could appreciate that bacteria present outside the necrotic core are mostly intracellular, being present inside the macrophage as indicated by the white arrow. This means that the bacteria are safe inside these macrophages and escaping from other immune cells. You could also see that green neutrophils interact with these infected macrophages, subsequently leading to the lysis of infected macrophages and release of bacteria as indicated by yellow arrow. Over the course of time, neutrophils lyse near these extracellular bacteria, as indicated in the panel showing 300 minutes by red arrow. So what all these mean for infection? That means the neutrophil interaction followed by infected macrophage lysis leads to release of bacteria outside, which now makes it more accessible to immune cells for infection control. Now neutrophils will lyse near these extracellular bacteria and it releases antimicrobial proteins that I mentioned earlier, leading to infection control. So this means that neutrophils are good for the host and control infection. But we need more solid proof for host protective functions of neutrophils. So how did we prove that? Next slide, sir. So what we did was we removed a gene named LTA4H, which is responsible for neutrophil chemotaxis, that is migration of neutrophils to granuloma. Then we infected these fish with Mycobacterium marinum and verified that neutrophils are mostly absent in these LTA4H knockout fish granuloma when compared to the wild type control. Then we also observed that these granulomas lacking neutrophils have very high bacterial burden, thus confirming that neutrophils are good for the host during TB infection. Next, we focused our efforts to understand the type of cell death pathways triggered in these neutrophils and interacting macrophages. So different types of cell death pathways exist in nature like apoptosis, necrosis, necroptosis, pyroptosis, ferroptosis, etc. So to identify specific cell death pathways triggered in neutrophils and macrophages, one should look at the transcriptome of the specific cell types to get clues about the triggered cell death pathways. So how will you do that? Next slide, sir. So to achieve this, we performed single-cell RNA sequencing with our granulomas. So in this technique, what we do is the tissue of interest, and in our case, the granulomas, will be dissociated to get individual cells, and then the total RNA from each cell will be separately captured and sequenced. Next slide, sir. So the final data will look like this, where individual cell types colored differently can be identified and grouped together based on their mRNA profile, like macrophages, monocytes, neutrophils, etc. And you can select your cell types of interest, in my case, neutrophils and inflammatory macrophages, and can further look for cell death associated mRNA transcript expression in these cell types. Next slide, sir. So we did this analysis for our granulomas, and what we found was that, as indicated in the boxes, mRNA levels of pyroptosis associated genes were specifically upregulated in neutrophils and infected inflammatory macrophages. And these genes are mentioned above. They are caspase, gasdermin A, AC. Don't worry about the name. I will explain it in the next slide. So, but what the take-home message from the slide is that this suggests that these cells are undergoing pyroptosis mediated cell death. Next slide, sir. So what is pyroptosis? So don't worry about this complex pathway. I will simplify it for you. So this pathway gets triggered by an external stimuli like bacteria, virus, or toxins. And upon stimulation, it activates a protein called ASC, which further activates a protease called caspase-1. And this protease further activates a protein called gasdermin D, named here as GSDMD, which is a pore-forming protein. Upon activation, gasdermin D, that is GSDMD, form pores in the cell membrane, leading to host cell lysis. And this pathway, by lysing the cell, helps in the release of intracellular pathogens so that it could be accessed by other immune cells for killing. In my case, if neutrophils and infected macrophages are undergoing pyroptosis to control infection, as predicted from my single-cell RNA-seq data, Then removing the gene essential for pyroptosis in the host will lead to loss of infection control. And the granuloma in these knockout fish will mimic the granuloma lacking neutrophils with very high bacterial burden. Next slide, sir. And we observed this to be the case when you knock out the gene ASC that I mentioned earlier from the fish, which is important for pyroptosis. You could see that the granuloma from ASC knockout fish exhibited very heavy bacterial burden. So this says that neutrophils undergoing pyroptosis is essential for bacterial control. Next slide, sir. So based on all the data that I presented, I propose a working model as follows. So neutrophils and infected macrophages interact with each other in a granuloma. This interaction results in lysis of infected macrophages and release of intracellular bacteria. How? Based on published reports, we speculate that DNA from neutrophils released during netosis, a process that I mentioned at the beginning of my talk, may be involved in this mechanism. Then, these extracellular bacteria could release products that could trigger cell death in surrounding neutrophils. This leads to the release of antimicrobial peptides from the neutrophils, which prevent bacterial growth, leading to infection control. So in a nutshell, neutrophils are good for the host during TB infection. So how can we use this study for better TB treatment? From this study, we understand that neutrophils can reach the infection site very effectively and can release its contents near the infection site. So if you could make neutrophil as a drug carrier, an idea which is currently being researched a lot, you can kill the bacteria more effectively in less time, possibly reducing the treatment duration for TB from months to days. So with this, I am ending my talk and I would like to show my next slide, sir. And I would like to show my fish facility where we grow all our experimental fish. And next slide, sir. Uh, and this is my beautiful campus with a big chapel at the center. And next slide, sir. And these are my lab members, and I would like to thank all of them for the help, and special thanks to my boss, David, who is sitting at the center, for funding me to do this work. And I'm happy to take any questions. Thank you.
Participants, it's your time to unmute yourself and ask questions to Dr. Gopinath sir. Participants,
Sir, good morning,
participants.
sir.
Good morning. Good morning.
I have done, sir.
Please proceed. Please proceed.
Sir, basically, this is an immunity-dependent disease, sir.
Yes, it's an immunity dependent disease.
So, why can we try alternate immunity or artificial immunity to treat this?
What do you mean by artificial immunity?
We know, sir, that stem cells can differentiate into any kind of cells. Why not we try that stem cells to be T cells, B cells, all immune kind of cells to treat this one?
So actually in this disease, the immune cells go to the site and contain the infection in most cases.
Yes, sir.
So that's not an issue. Only in some cases where maybe some genetic mutations that may affect the host immune system, which we don't know what kind of cells are affected, that causes problem actually. So once we know, we can think about if we could use stem cell therapy or anything to address this issue.
Okay, sir. Thank you, sir.
Yep.
குட் மார்னிங் சார்.
Good morning.
சார் இப்போ வந்து நம்ம நியூட்ரோஃபில்ல வந்து ட்ரக்கை வந்து லோட் பண்ணி நம்ம அங்கே அனுப்புகிறோம்னு சொல்கிறீங்களே சார், சார் இருக்கும்
புரியல என்ன?
மற்ற செல்ஸ் அஃபெக்ட் பண்ணுமா சார் இந்த ட்ரக்கு?
இல்ல, ட்ரக் வந்து சேஃப் தான்.
ஐ மீன் அதோட
சேஃப். ஸோ ட்ரக் ஆல்ரெடி சேஃப், எஸ்டாப்ளிஷ் ட்ரக்ஸ்.
ஓகே சார்.
So just a hypothesis, neutrophils drug load பண்ணி அமிச்சா maybe we can speed up. So nobody has tested, it's just an hypothesis.
Okay sir.
Yeah.
Any more questions? Dr. Gopi sir.
சொல்லுங்க சார்.
I found a question in the chat box.
Okay.
It was asked by Miss Akshaya, and she asked whether neutrophil act as better treatment for only MOB. Mycobacterium tuberculosis. Yeah.
You mean making neutrophil as a drug carrier act only for MTB?
Yeah.
Not. There are some research going on to in cancer field where they are trying to make neutrophils as a drug carrier to target cancer cells, but these are all in very early stages. That is, they are not at all even in the clinical stage. They are all in the early stage. So I just put that only to just to say that some application for my research. That's it. Proposed application.
Thank you, Akshaya. Any more questions from the participant side? Participants, any other questions? Okay. Hope no other questions from the participant side. Okay, Dr. Gopi sir. I think
Okay, sir.
No more questions from the participant side.
Okay.
I hope this is the end of your presentation, I think so.
Yeah, it is my end.
Right, sir. So, we thanks a lot, Dr. Gopi sir, for accepting our invitation and spend your valuable time with us.
Thank you, sir. Thank you very much.
Thank you so much. And Dr. Prabakaran sir.
Yes, sir.
Should we move on to vote of thanks?
Yes, sir. Yes.
So I take this opportunity to invite Dr. Prabakaran sir to propose vote of thanks.
Thank you. Honorable correspondent, principal, chief guest, head of the departments, present excellencies, staff members, and my dear young minds. I feel immense pleasure to propose vote of thanks on this occasion. First of all, I thank the college for giving me the opportunity to express my gratitude on this special day. It is the biggest day in the history of our Ayya Nadar Janaki Ammal College that the Department of Biotechnology hosted the 5-day international webinar on recent advancements in cancer research and therapy. Since 1963, the institution providing higher education to strengthen the rural areas in the southern part of India. Here I want to emphasize a concept given by our former president, Dr. APJ Abdul Kalam. He started a scheme named PURA, which means providing urban amenities to rural areas. In the same vein, our correspondent, Thiru Abiruben, and our principal, Dr. C. Ashok, had a vision one step ahead, that is, providing international amenities to rural areas. We all are honored to be a part in your eminent vision, sir. I thank for your continuous support. Next, I extend my sincere thanks to our beloved HOD sir, Dr. M.K. Rajan. He is always engaged in to uplift the students' curriculum. Whenever we approach him, he spares his valuable efforts to bring best out of us. In fact, he is the backbone of this event and took all the responsibility and guided us towards a grand success. My special thanks goes to IQAC Coordinator Professor R. Jagannathan and Deputy Coordinator Dr. Senthil Kumar, Dr. Jabakumari Gulla Vasanti Madam for their encouragement which make us to conduct this kind of events. As no program can become successful with a single person, here I extend my sincere thanks to the faculty members of biotechnology, Mrs. Sugapriya Menaga Madam, Mrs. Geetha Madam, Dr. Muthukrishnan Sir, Dr. Pandiarajan Sir, Dr. Selvarathinam Sir, Mr. Mageshwaran Sir, who directed this event from day 1 to day 5 in a smooth manner. Last but not least, my heartfelt thanks goes to my beloved mentor, Professor Samuel Pondian and Dr. Vijayakumar Sir, the two co-organizing secretaries of this event, who took all the responsibilities on their shoulder and supported me to organize such a challenging event in a short span of time. Last, my sincere thanks goes to Mr. Venkatesh, head of the Department of Visual Communication, who provided all the technical support to reach out this event globally, even in this pandemic situation. Also, I thank the lab attenders, Mr. Palani, Mr. Ramkumar, Mr. Andumani, and the lab assistants, Mr. Vadivel, Mrs. Yuvarani, for their valuable efforts. I would like to thank all the people who directly and indirectly supported us throughout the event. On behalf of our institution, I would like to thank the gems of our event, so-called our eminent speakers, Dr. Balaji Chandrasekharan, Dr. Tyagarajan Ramesh, Dr. Ashish Tyagi, Dr. Senthil Kumar Chingu, Dr. Gopinath. I am proud to say that we blessed with a reputed speaker across the world who spared their valuable time to enlighten our participants. I believed that their presentations are well received by our students and our other participants across the globe. I hope this event inspired many students and given a thirst to take up research on cancer biology and infectious disease. I thank all the student participants from our college and also from other institutions across the globe. Thank you.
Thank you, Dr. Prabakaran sir. Thanks for your nice vote of thanks. And now we have reached the end of the session, and as we have planned, we have a small discussion with postgraduate students. Postgraduate students of Department of Biotechnology are ready to interact with the experts who have presented their expertise field with them for the past 4 days. So now now this is the time exclusively for the benefit of the students, right? So as our beloved principals are directed, we have planned this session to facilitate you with interact with the experts of the program. So they are ready to interact with you and they have spent their valuable time with you because they are seeking your most uh, interaction, right? So don't waste this time because it will not come again. Okay. So arranging this kind of session during this pandemic is not a easy thing. That is known to you. Please consider it. And moreover, all the experts are in a same coordinate. And as Dr. Prabakaran sir said, Dr. Balaji sir is with us, Dr. Tyagarajan Ramesh sir is with us, Dr. Ashish Tyagi sir is with us, and moreover, yet another expert from the zoology department, Dr. Lakureddy is also with us. He is from University of Texas, and moreover, my students, Dr. Prabakaran and Gopi, they are also with you. So this is the time for you people to interact. So open your mind, ask your questions, and get clarified. Thank you. The floor is yours. Participants?
Sir, excuse me, sir. Senthil sir joined, sir.
Sendil sir, illa Dr. Prabakaran sir.
Okay, sir. please.
செந்தில் சார்
I
இல்லை
will call you.
சார், பாக்கி சார்லாம் இருக்காங்க. ஆஷி சார் இருக்காங்க, பாலாஜி சார் வந்துட்டாங்க, கோபி சார் இருக்காங்க, ரமேஷ் சார் இருக்காங்க, அண்ட் ஃப்ரம் எக்ஸ்பர்ட் ஃப்ரம் சுவாலஜி டிபார்ட்மெண்ட் ஆல்சோ இந்த ரூம். Students.
Sir, good morning, sir. Sir, first of all, thanks for this opportunity. And is that mandatory to clear TOEFL and IELTS for abroad PhD postdoctoral job, sir?
Not necessary. This is Dr. Lakureddy from Texas. For the PhD, you may or may not need it. Depends upon the university where you are approaching, and they ask for a GRE. Depends upon the where you go, and many universities may not need that one. Since you finish master degree, going for a PhD, they may not ask you to do that. But if you have cleared the TOEFL or GRE, that's going to be plus, and you may be able to get the funding immediately. So, and that will be additional help for you.
Thank you, sir. Thank you.
Samuel, sir, if you don't mind, can I present for a few minutes, 5 minutes of time? So the students still there, and they need to know something from my side too.
Sir, please proceed.
Make me as a presenter.
Sir, the presentation is open to all, sir. There is no restriction.
Okay. Are you able to see my slide, sir?
Sir, no, sir. I couldn't. Participants, student participants. Participants, please post your questions because the experts are waiting. Okay. Sir, very good morning to one and all. Dr. Balaji sir, Dr. Tyagarajan Ramesh sir, and Dr. Ashish Tyagi sir, and Dr. Gopi sir. Very good morning to one and all. So what we have a plan is to establish a network between the students community of ANJAC with you people. So for that, what we have to do, sir? What are all our requirements? What we should do, and what are all the expectations from universities of abroad? So can you please?
Are you able to see, sir? Samuel, sir?
Yes, sir. Yes, sir. Yes, sir.
Okay. So my name is Lakureddy Alagarsamy, and all the publication, if you put it AL ready, it will come. And more than 22 years, I did cancer research since the Cancer Forum. And yesterday I was talking to Dr. Ashok, our principal, sir. Then he said that why don't you participate in this series? And I knew Vijayakumar little bit through some other connections, and he did PhD under my supervisor's wife. So currently I'm administrative director and senior clinical manager. From the bench research, I moved to the clinical study. I'm carrying clinical study in nephrology, cardiology, and GI liver. And I also involved in the cardiology fellowship program for the research guidance, as well as we run the internal medicine students. So like our M.B.A student, we run this program. I am the advisor for the any clinical trials or clinical research also. And I would like to thank the Anjak because I started from there and I got a confidence from the Anjak college only. I'm not saying for the sake of presenting here, but this is my standard slide. Wherever I go, I start with this slide because I never grow without the support of many hands. That's what I start with it. Another thing in Tamil, I need to say, Sakanda Gupumil or Karpu Vricham and then I will start. So reason is that that's the place I got a confidence. That's the reason I got the support to go next level. And I did more than 20 years in cancer research. Then I move on to next level to study the clinical trials. And because all the experts from here, all our friends know that how the cancer cells survive. In general, if you look at the left side and you see that Sprouty and Sybil, this is a negative regulator receptor tyrosine kinase. In normal pathological condition, these two genes act as a brake and it'll put the downstream signaling to be under control so that cells normally grow. But in the cancer stage, these two genes will be lost because this is a proto-oncogenes. They'll be lost, and then obviously receptor tyrosine kinase induce a lot of signals downstream. Then transformation, cell signaling happen, then cell growth uncontrollably. And so in cancer, in terms of cancer, when we are talking, the people are talking, when we are started the cancer research, the cancer kills quickly. That's what believed the people before. But through the research and new invention, now the cancer, especially breast cancer, is like normal, like any other diseases. And particularly when they found the many therapies to cure the cancer, and particularly I want to take this as an example. In the non-small lung cancer, the EGFR mutation one of the culprit to kill the person very quickly. Any patient has the EGFR mutation, they are not responding to the therapy. Initially, they treat that EGFR is the causative for the cell growth, but later they find that EGFR antibody is not effective anymore. Then they find out EGFR mutation is the culprit. So some of the non-small, 15 to 20% of the non-small cell lung cancer as this mutation, they can survive. So they have to identify different mutation-specific antibodies. So even though many research is still happening, and there are so many antibodies, so many cell-specific therapies and other things available, even stem cell therapy is also available, but still a lot more needs to be controlled in order to avoid or eradicate the cancer. I don't think it's going to happen, but we can slow down the process and we can maintain the longevity of the cancer patient, and then that's what happened. So even my research is like, well, since I said I worked in two different genes, Proudy and Sybil, the my good connection people may remember that last year 2019 Nobel Prize winner Dr. Greg L. Simmons, he did find this HIF1 alpha is the hypoxia-induced vector 1. That's a gene can induce that can help to survive the cancer cell in the low hypoxia environment. So I'm happy to share here as well. I had a good connection with him. I also found that my Sprouty gene negatively regulate the HIF1 alpha. When it's a normal gene, the normal cells, the Sprouty one is there, HIF1 alpha never express. But in cancer stage, Sprouty is gone, then HIF1 alpha is expressed more. That can give the cells to grow, the cancer cells to grow very faster. Not only that, I have a trainee called Dr. Pallavi. I trained her and she worked with Dr. Alex Simmons. This is the notice I received from associate get the Nobel Prize. I congratulate him. Immediately he responded, even though you know that how busy it's going to be. So I had a great connection. I know that I worked more than 20 years for the cancer research, and I started with my PhD also on cancer research as well, particularly in cervical cancer. You may able to see my slides, or it may not be. I can tell you one thing. During my thesis, and I put the cover page saying that even though we spend a lot more money on cancer research, but still we did not reach the goal where we need to be. Even till today, you see that number of amount spending for the cancer is very enormous. Any foundation collect tons of money from the people to treat this cancer and that cancer, but still we are not able to reach the goal what we want to be. And so this is like a current slide from the American Cancer Society. If you see this, nearly 900,000 males are affected by cancer, and 900, but the females are more affected than men. You can see this here. Even though if you ask Senthil or Prabakaran or anybody's living in USA, they know that amount of spending for the cancer research is enormous. Anybody get the grants and cancer research. That's the reason many private universities working cancer research. For an example, when the president for the infectious diseases in America, they may not spend that much of money. They ignore them. They're saying that, oh, America doesn't have any infectious disease. But if you see the cancer research, like enormous, huge amount we are spending. Still, you see the number of cases here. We are talking about the COVID and 250 deaths only, but still crying for the political reason also. But if you look at the estimated cancer death in 2020, this year alone, 320 male is already lost their life, and 285,000 female is lost their life. So still need to be done a lot more. And there are many young minds like you guys working on. You need to come to the research. The reason I'm saying that even I came from the Sivakasi College one day. So those time biotechnology is a growing. But I worked in a zoology department. I studied in a Madras University PhD, and then move on to All India Institute of Medical Science. I happily worked with Dr. Indranath. He worked as an infectious disease person also. If you look at the leprosy research without Indranath, now leprosy research in India, he is the pioneer to bring the biotechnology terms to India. I'm happy. I'm worked with the 2 years, and I then moved to Harvard Medicals to do my postdoctoral research. And 13 years I worked as assistant professor in Northwestern Medical School in a sprouting field. Then now I moved to clinical side. Reason I moved, because whatever we do, the bench research is not directly connecting to the patient. So I want to make sure that I have a connection with the patient to treat. That's the reason I move on to the clinical trials area. So now I'm successfully running many different trials. I'm a co-investigator as well as co-founder for the several organizations as well. And I'm leading many channels. I'm editorial board member as well as reviewer for the cancer field. Still, I'm continuing to do that. And with this one, I want to thank everybody for the. Yesterday, I spoke to Dr. Ashok, our principal. He told me that why don't you participate and lead this conversation? Our student need that. As Samuel said, as you asked, what are the connections you wanted? This is very simple, sir. We can have many things to help it, and many university students like young minds, we need it. So America, one of the American education system, there are a lot more students needed from abroad. They cannot run this America without our contribution like people. Everybody may agree. And Gopinath, he know that he's in Duke, and he knew that without our minds, this America cannot function. The reason is that the American family system is broken. After once they celebrate high school graduation as a bigger manner, because that's the level they can reach it. After that, because they need to spend tons of money to get the graduation, medical degree and everything, so people may not go for it. Without us, without mind like young brain, this country cannot function. You need to come legally. Don't approach to anybody illegal person. They say, oh, I take you to the American thing. Do not believe any of them. Go to usgovernment.org and you get all the tons of information. You'll get the guidance, because this day you are spending all the time in your gadgets. Find out some good information. I'm feel free to help anyone want to come for abroad for the PhD or postdoctoral fellow, whatever the level you want to come. I'm happy to help you. I'm happy to guide you. I do regularly help people, and I got help from many people. That's what I want to say. Same thing I'm proposing here. If anybody want, I studied with M.K. Rajan sir because he took biochemistry for me when I was doing master's degree in zoology department. I was the batch in 91, 93, and still I have a connection with our department. I'm happy to say that our Bhaskaran sir, our late Bhaskaran sir, is my master degree project guide. And I worked in Delhi. I came to see Bhaskaran sir. I provided 2 micropaper to the Bhaskaran sir. Maybe still the paper is in this your biotechnology department. With this one, I want to thank you for the opportunity. Definitely, students, please use this opportunity. You may not get this kind of opportunity. At least 5 students get out from this one. Will be very great success for your department. And people like Gobi, Palaji, Ashish, many people may be willing to help you. because you need to reach them. So I always, whenever I go to any talk, I always raise my hand. People say that, oh, Reddy here, then he may not be stopping. You need to ask the question. If you don't ask it, you may not be able to get out, because no question is a stupid question. Because you do not have an answer. That's the reason you're raising the question. I never stop it. Any meeting, even nowadays, any meeting I go there, my hand is first. I never stop them. This started in our M.Sc zoology department. Our late Albert Mohandas sir always encouraged people to ask the question. He give you moral value talk rather than giving lecture about the topic. You can go and read it yourself. You don't need a staff to give the lecture for 2 hours to you read it next day. That's what he tell. I believe I got a lot of support from them. Even Bhaskaran sir is a great kid for me, great mentor, great father, great teacher for me. I got a confidence from there. That's the reason I happily say that those who are living in the Sivakasi area, they know that what I mean for Kandaga Bhumi. Very hard. Very hard. But still, I was able to succeed from there. People say that Anjak is a jail, but I learned my education from there. Even Dr. Ashok says the other day, he admitted, still today they are saying this is jail. Any person can learn. Even many people from jail, they came out very good. Even Gandhiji, even Mandela. They spend their time in jail, but they learn a lot of things. They lead the country. This is the opportunity, friends, young minds. Please get out of your cocoon. Ask the question. Approach everybody. You have to realize who you are. No one can help you without asking question. No one can help you if you don't identify yourself who you are. I can tell 1,000 things, but if you don't realize that who you are, you cannot come to the next level. Please, this is your time. This is your chance. Identify yourself. You ask yourself who you are. If you don't self-evaluate by yourself, you cannot succeed. Hard work, confidence, successful. Come in the next line. If you don't have the two things in the first place, you cannot imagine for successful. With this one, I conclude my thanks. Thank you for the opportunity. Thanks for all the friends that did a good job. I really appreciate this opportunity. Thank you, sir. Thank you for taking this place.
Sir, I have one more doubt, sir. Sir, what about a program cell which can or
Excuse me. Samuel sir, please mute your audio. Samuel sir. Nishant, please proceed. Please proceed.
Sir, what about a programmed cell or a pre-designed cell which can withstand or fight cancer? Is there any possibility, sir?
Can you repeat the question?
I'm asking that, sir. What about a programmed cell, sir, we can program a cell which can withstand or fight against the cancer cell, sir? Is that possible?
Yes, possibility. There are tons of research going on the program cell to deliver specific spot to change it. The problem with this one, each cancer is unique and each individual is unique. Whatever you do globally, you cannot control. That's a problem.
Sir.
Okay, so the
sir,
uniqueness.
I have one hypothesis.
Yes, sir. Go ahead, please.
So we all know that induced pluripotent cells can convert and differentiate into any kind of cells, and we all know that white WBC cells are only withstand till now cancer cells in innate immunity. Sir. Why can we try to that? To convert that induced pluripotent cells into NK cells or T cell, B cell with adrenal injections, which can produce a hyperactivity to act against that cancer cells, or we can induce that NK cells or B cell, T cell in our body as the innate immunity. We can induce that innate immunity, sir.
Brother Balaji, brother, you want to take the share or you want me to continue the talk?
Sir, I think I am done, sir. This is my hypothesis, sir.
Okay. Yeah, so there are many in cell therapies like a pluripotent cell induced cell therapies available. The problem with this in delivery method and this cell type where it's going to be, because they may mislead to the different direction. For an example, in cervical cancer, in normal cervix is the squamous cellular squamous cell type. But when the cancer normal cervix become a cervical cancer, the cells modified into glandular cells. They start secreting mucin-like protein. They modify the coat. The cells, like they change some complete structure. Whatever you want to deliver to the mechanism through the pluripotent cells, they may identify the original, but not the modified cell. That could be my vague theory, but I'm not giving any specific answer to you. That could be one of the good answers, but you can read it through the internet. You can Google it. You can find some more information. Suppose if I find something, I will send it to you in your way through Dr. Samuel, sir.
Thank you so much, sir.
But definitely don't stop. It's continue to read, and that's the only thing give you the next answer and reach to the particular person.
Yes, sir. Hello, sir. Yes, sir. Hello, sir. I have a doubt. So, what are the qualifications we need to improve and perform as a JRF or SRF as well as a PhD program in abroad? Otherwise, what are the qualifications they expect from us? Any idea, sir? Suggest us.
Sir Samuel, sir, please mute your audio, sir. Background voice is disturbing. Yeah, please.
சொல்லுங்க சார். Yeah, I will reply. Yeah, I will. So, first of all, I would like to thank Dr. A.L. Reddy for giving such a very nice advice for students. When I was doing my M.Sc, I never got that kind of advice from anybody. So these students are fortunate to get advice from such a very nice person. So there is no such thing as JRF, SRF in abroad. I mean, in US at least. So at least as far as my university is concerned, so what I see is if they are taking people from outside their country, outside United States, one thing they look at them is sometimes they look at if they have any papers as as a second author or third author or something like that. So they look for some 1 or 2 year experience as a project scientist or project student or somewhere if they join something like that. So that will help. I don't know much about TOEFL and GRE because I did my PhD in India. So in addition to what Dr. A already told, I wanted to add one more thing. In addition to looking at abroad, you can also look at very good institutes in India. They also provide very good PhD programs. They are not inferior when compared to programs in United States, or because the postdocs who work here are trained in India actually. Actually, India is giving workforce to United States. So we are all very well trained in India, and then only we go to US and then we pick up here. So you should also look at programs in India, and for that you should start preparing for CSIR. And I don't. My time it was CSIR only, and also some other exams. But I don't know about now the situation, what the situation is. But whatever it is, you should use this COVID situation. Maybe the college, I don't know whether the colleges are running promptly from 9 a.m. to 6 p.m. But if you have some extra time, read a lot of books like Leninger, biochemistry by Leninger, cell biology by Bruce Alberts. So these kind of books will help you in clearing exams in India, because since I cleared exams in India, so I am telling you my experience. So you can use this time to prepare for these competitive exams. Study a lot of books other than your syllabus. That will help you a lot in getting very good institutes in India, like IAS, Indian Institute of Science is a very good institute. IITs, all the IITs are good, so you should try getting into those kind of institutes. That also will help you. So in addition to abroad. So that's my advice.
Yeah, my experience, Kobi sir, well said, and this is correct. And even India, we have like a well-reputed people, well-reputed institution. Even infrastructure is more better than any of the local institute here. It's nothing lower than that. Even I worked in All India Institute of Medical Science. I had all the opportunity, all the infrastructure, what I need. When I come to Harvard Medical School, I find sometimes it's difficult because the department to department we cannot travel. But in India, there is no restriction. You can go to any department to get any kind of help. But here, one floor to next floor, you may not be able to interact each other because each and every penny is count for the accountability for the bosses, because they want to survive with their own money, because they need to fight to survive themselves. Yeah, they may not be able to provide it, but in India, you have a very good reputed institute, well-known scientist, you can work with it. If you want to study in abroad, what you could, as the Kopi said, is the correct information. You go to MK University, you work in a biotechnology department or any other department for 2 years as a research assistant, get 1 or 2 publication, then come to here. But if you instead of doing this one, if you do the PhD, the coming here is the chances are more. And if you come as a PhD student, you may not be able to get 100% scholarship. Then you have to end up paying a lot of money to the America. That's in my view, that's not wise decision, unless otherwise your parent has the tons of money to provide a support. Go there, even CSIR Institute in Delhi, all the major cities, any institution, any reputed institution, go and involve yourself. In my experience, what I did, when I was doing a master degree, I cleared GATE examination that time, because that's the first time they said the GATE can be eligible for PhD. And that's the thing I cleared before my master degree examination. That's a green card I got it to go to the research. I did not spend any money from my pocket, and all the money provided from the central government. I got like a full scholarship for like 4 years of PhD. Then I moved to All India Institute of Medical Science. Then I got an INSA award. We have tons of awards program. You need to be constantly looking at it. You need to approach them, and you need to have a vision to keep on knocking to this one. You will get the money there. So I think the great opportunity. Even nowadays, there is even 8th grade students also getting money to go to the higher education. But nowadays, there's a lot of money government is providing. Please utilize them. Get the research from local government.
Thank you, sir. I'm really happy and impressed. And I have another question, sir. One of the British scientists have approved as use human cloning techniques to perform embryonic stem cells collection. This was a relaxation in some of the countries, but not at most in India. For that studies, we can choose in abroad, sir. So what we do for that?
So any any targeting any specific study, you need to work in a cloning area. So without experience, they think that coming to America, people think that you are coming as an immigrant. You have a intent to come and stay in this country. Unless otherwise you show like who you are really interested in the cloning technology, you have an experience in the cloning technology, then only they can allow you to come here. Without that, they will say that but you can reach to the particular institution, keep on writing in the email because they may not read it. But when some of the people like me, I always read any email. I don't ignore them. Any text I get, I don't ignore them. I send a message. At least I'll say thank you. Then I'll reach out to you when I get an opportunity. But you need to keep on knocking. Without knocking anything, it won't come to your hand. So you need to pursue which line you want to be. And definitely, I hope you have very good interest in the research. Continue to read, continue to reach the local lab where you can work for 2 years, at least get some publication, then move on to the any places what you are looking for. Hope this
Okay,
address
sir.
your
Yes, sir. Yes, sir.
very good.
Thank you so much, sir.
Yes, sir. Sir, I'm not a such an experienced person, sir, as compared to others. But just to work hard, one day everyone's going to succeed. That's it. Just work hard. That could be push to go forward. That's it. My simple advice. That's it. Thank you.
Excuse me, Dr. Ashish, your voice is not audible.
Can you hear me now? Yes. Yeah. So thank you so much, Dr. Samuel, for inviting me and giving me this opportunity. And I think Dr. A.L. Reddy has already given every advice possible, such an impressive talk and such an encouraging talk to the kids, to the students. I would say that the kids, the students in M.Sc, they can just start preparing for CSIR in their M.Sc itself, CSIR and ICMR exams, and it's a good fellowship for PhD. Earlier in my time when I cleared CSIR and everything, it was around 8,000 or something, but now it has increased to, I think, 24,000 or something. So it's a good amount to start with. It's a good fellowship. And anywhere in India, you can, in the good CSIR institutes like IGIB in Delhi and other institutes, very good institutes, and ICMR also. In cancer research also, there are institutes in Noida which are doing wonderful work. They are publishing in Nature and Cells, Cell Metabolism, and every good peer-reviewed article are being published from India. And students who have cleared ICMR and CSIR exams are publishing in those journals. So they give you a good start, and as soon as you finish your PhD, and from those institutes, you are bound to have good publications, at least one or two first of the publications, one publications at least, and then you can try for PDF here. that would be an ideal way to come to you. As in my point of view, although Dr. A already has and Dr. Gopinath has already told you, but PDF positions after getting a CSIR fellowship and or ICMR and other fellowships also. DST is also providing fellowships, and ICMR, CSIR both have PDF fellowships also. In those cases, you have to approach the institute where you want to do PDF and with a scientist, design a project and then submit it to ICMR so that you can get an admission as a PDF or a research associate. So there are several venues open for everybody in India for the PhD program, and the next step would be a postdoctoral fellowship in not only in US but all over the world. In Germany also there are so many good institutes. In Belgium, Ghent University, and those institutes are very good doing cutting edge research, so they can apply there also. And yeah, so the first step would be to prepare, and as Dr. Balaji said, work hard and getting a position where you can apply, and people are ready to take you as a research fellow in their laboratory. And yeah, and thank you so much for inviting me, and thank you so much for giving me this opportunity.
Sir, we have a question in chat box.
Okay. Sir, a question. Sir, I have a question which was asked by Ms. Gayatri from second M.Sc. Biotechnology. The question was, can a programmed cell act along with normal cells in a tissue after stem cell therapy? So the question is, can a programmed cell act along with other normal cells in tissue after stem cell therapy is done to a cancer patient? Dr. Ashish, sir? Dr.
Yes,
Ashish?
sir. Can you please repeat the question? I could not.
Yes, sir. I'll repeat, sir. Can a programmed cell act along with other normal cells in a tissue after stem cell therapy done to a cancer patient?
So it can be done, but there are several issues, as Dr. A already pointed out, that during programmed cell differentiation, there are a lot of problems which occur, and there are some topics where programmed cells are being used, but it is still a topic of debate whether they can be used with the normal cells during stem cell therapy. But they are being used. It is being researched, but still a controversial topic. But yeah, people have reported both the stages. People are using and getting positive results. People are using which didn't get the positive results. So yeah.
Yeah, I want to add something along with an STI because I don't want to give myself and all the questions as well. I want to give the opportunity to everybody. And so there are occasions that people tried the question raised and the future what they find at a later stage, and then whatever the pluripotential given to the patient, they move on to the not only the cancer area, they move to the normal cells and they form the colony, then they become the problematic in later stage. even an animal model, and they find, for an example, if you target for the breast cancer cells, and they because this is the glandular cells, as endothelial cells. Wherever endothelial cells are there, they go and form the colony in a different place. Then that became a problem. Not only breast cancer, you are curing for breast cancer, at the same time you are inducing another type of side effect in the different places. So there's both pros and cons, but it's not 100% pure therapy for any of them. As I said, in a unique situation, every individual is different. Every cancer condition is different. Based on the immunity of your patient and everything, it varies and it forms a different thing. But most of the studies in animal model level, and some of them has been tried in cancer patient also, but still it's not 100% success, I can say.
Participants, any other questions from your side?
Sir, one more question in chat box.
Sir, is it possible to use stem cell to cure spinal cord injury?
Yes. And I mean, the stem cells, because since many of you know that this is a pluripotent cells, it can be differentiated to any kinds of cells, and even cartilage removal of the, like making the new cartilage. For an example, sports injury person will have the damage in the cartilage. Even they use the stem cell to cure the cartilages. Many different the same way they can use for the backbone also. But there's so many, but still the success rate is not that great. And there is another question from the chat box. I was reading the nanoparticle delivery. Yes, there are many nanoparticle delivery mechanisms for the cancer such as identified. Still at the cellular level, animal model level, this 100% success rate is shown so far. Even for an example, even EGFR mutation genes also, they find the delivery mechanism through the nanoparticle. But still, the success rate is not 100%. That's the reason none of the mechanism, what are the cutting edge research we are doing it so far. Still, it's not 100%. The cure for the cancer is not identified or any other diseases the same way. And but definitely, nanoparticle delivery is little more specific than any other as well.
Dr. Prabakaran, sir.
Yes, sir.
You please go ahead.
Yeah. Dr. Reddy, sir, great applause for your speech. Not only the students inspired, including all the fellow men, we all inspired by your talk. Really, I don't know that you are joining today. It's a real great surprise.
No, yesterday I was talking to principal and Ashok sir, because I was doing M.Sc. He's the young faculty joined to the team, our college, and he was our subwarden in the hostel, postgraduate hostel. And so when I was giving the talk, and he came and sit there, and he was talking. Then yesterday he called me, said that there is a cancer talk is going by technology department. MKSRs are conducting, so I will ask them to include you as a discussion panel. Please go ahead and do it. Even morning he texted me that I was going to MK University. Please join the discussion panels. I said definitely, sir, I will join. And thanks to the Sevarkodian sir, he is the one sent me the link. And I know Sevarkodian also. When I was doing the first year, I think he is doing M.Phil there. And I have a good connection to all the departments there. And as I said, our founder, late Dr. Bhaskaran sir, is first HOD for the biotechnology, I believe. And Kannan is my junior, and he was working as a faculty at that time. Now he's in Pandiarajan University. I still have a memory, so still I have a connection. Even Ramakrishnan sir is in UG HOD. He was like much senior in University of Madras when I joined to the PhD program. Definitely. One thing I want to correct here. I never say this is my advice. I always say this is my experience. I never say to anybody that I'm giving you advice. I don't use that word at all. I don't believe in anybody saying that I'm giving you the advice. I don't like it. But I say that share the experience. That's what I always say. That somebody may have a different opinion, but I always don't use the word I'm giving you the advice. I'm giving you my experience. Whatever you want, you take it. That's up to you. So that's the thing. Thank you, Prabakaran sir, and thank you, Samuel sir, and definitely this team.
அதே மாதிரி ஃபாலோ பண்ணியிருக்க மாட்டேன். வெயிட் வந்து நீ வந்து குறைச்சிருக்காரு. இதில் பாருங்க, இது வந்து எவ்வளோ வெயிட்டாக இருக்காரு, அண்ட் இதில் வந்து எவ்வளோ வெயிட் கம்மியாக இருக்காரு சரி 10 தேசம் அப்படிங்கிறனால ஒரே மாதிரி வெயிட்லேயே வந்து நம்ம ஃபாலோ பண்ணி படத்தை எடுக்க வேண்டாம் அப்படின்னு சொல்லி அப்படி டிசைட் பண்ணி வெயிட்டை வந்து கம்மி பண்ணியிருக்காரு. ஓகேவா? ஸோ இதெல்லாம் வந்து எப்படி பாசிபிள் அப்படின்னு பார்த்தீங்கன்னா ஒரு விஷுவலைசிங் மூலமாக பாசிபிள். நீங்கள் வந்து இப்போ கூட கேரக்டரை வந்து இது பண்ணுறீங்க, கேரக்டரை வந்து நீங்கள் வந்து அதை வந்து எழுதிட்டீங்க. ஓகேவா? அது எப்படி எழுதுவீங்க அப்படின்னா ஒரு விஷுவலைசிங் பண்ணி தான் நம்மளால எழுத முடியும். ஓகேவா? நம்ம நிறைய கேரக்டர் சொல்லலாம். இந்த படம் படம் சொல்லுவாங்க. இப்போ ரீசெண்டாக வந்த படம் ராட்சசன் படத்தில் அந்த கிறிஸ்டோபர் கேரக்டர் இருக்கு இல்லையா, அந்த கிறிஸ்டோபர் கேரக்டர் வந்து சொல்லலாம். கிறிஸ்டோபர் கேரக்டர் வயசு கம்மியாக தான் இருக்கும், 70 வயசு. அவனை வந்து இப்படி மாத்துறாங்க இல்லையா, ஒரு வில்லன், ஒரு கொடூரமான வில்லன் அப்படின்னு மட்டும் வச்சுக்காம அதுக்கு வந்து அவங்க ஃபிளாஷ்பேக் இருக்கு. ராட்சசன் படத்துக்கு ஒரு ஃபிளாஷ்பேக் வந்து வைக்கிறாங்க. ஓகேவா? ஃபிளாஷ்பேக் வச்சு அதுக்கப்புறம் வந்து படங்களுக்கு மட்டும் கிடையாது.
Not to study what you are doing for. So at this stage, when you are a young student, you need to learn the techniques, RNA techniques, tagging, fluorescence tagging, how to track it, because that has a tons of technology involved. This is microscopic, because I know how much time you spend it, because I spend a whole lot of time in fluorescence tagging and everything. You cannot get succeed in one experiment. You need to do multiple experiments. So the technique, you don't listen to it because it is a cancer drug, nothing to do with infectious. No, the technology is the matter. What kind of technique he is using? This is the same technique used in the cancer research as well. The model disease model is taking differently, but the technology is the same thing. So please, students, don't ignore any availability or any opportunity available in any kinds of department. You can join to physics, biology, general biology, zoology, botany. Even I can tell you, one of the Vijayakumar college student, he did botany department there. Right now, he is a leading scientist in cardiovascular disease. He is the botany student. Dr. Saktivel Sadayapan. Anybody can type it. Because nowadays, you don't need to tell anything, everything in the Google. Please type it, Saktivel Sadayapan, Cincinnati Medical School. He's from Virudunagar. He's from your area. I am from Lathigalam. I never been a good student in school. I never had a first rank, mediocre student. Even I proudly say that I am a backbencher. I can reach to this level. You have the same opportunity here. Use it, utilize it. Because how much time you spend studying and reading and connecting to the people, that's the one take you to the next level. Because this is previously they used to say that don't spend more time on your like a mobile, but this is time COVID brought everybody to the mobile technology. But use it wisely, what you want to see it. Because that mobile has everything for you. The way you use it, way they utilize it, that will take it to the next level. So you need to have a goal. For an example, if you want to go to the Madurai, you need to get the right bus. When you go to the Sivakasi bus stand, you have tons of buses there, right? You have many different directions. But you need to choose, when you want to go to Madurai University, you want to choose the Madurai bus. Same way, in mobile also, please. You choose it what you want to do it. Then go to the mobile. Spend time and learn and go to the next level.
visualizing
Thank you.
most comes. Sure, pleasure.
Thank you, sir.
In everyday life, next class
Thank you so much, sir.
Types of image
Dear participants, any more questions? So hope we will reach the end of the session. Dr. Prabakaran, sir,
Yes, sir.
should we conclude the program?
Yes, sir. Please proceed. One more thing. I want to have a humble request to Dr. Reddy, sir. In upcoming event, please share your views, sir. We will approach you.
Anytime. As I said, I can share my number. Anybody can. As I always say that I never ignore any emails. I never ignore any text and wherever I received. And definitely I take time. Even here also, as you know, we had to work more than 12 hours of time. Otherwise, then only you cannot survive. My sleep time is 4 to 5 hours only. Even Gopinath and Palaji and D.Y.Ed can accept it because they know how they spend the time.
Yes, sir. Dr. Senthil Kumar Singhu is my lab senior.
Wow, that's okay. Where?
In Korea. He worked in Korea.
Yeah,
Yes.
Yeah, I think he worked with the Dr. Niranjali Devaraj. I'm the student with the Dr. Devaraj. He's the University Vice Chancellor. I only converted his lab into their lab is doing toxicology. I'm the first student convert his lab into cancer research. When he got the Vice Chancellor position, he called me 3 a.m. in the morning. He told me that you want to take it to the next level. Yeah, thank you. Yeah. That's what he always said. Very simple person.
Thank you, sir.
Thank you.
Sir, please conclude, sir. No, sir, we can wind up. Yeah, because it's midnight for them. Please consider that. Yes. It's midnight for them.
Thank you so much.
Thank you, sir.
Thank you, sir. Thank you so much.
Thank you, sir. Thank you, sir. Thank you, sir.
Thank you. I thank all of my. Yes, thank you.
You answer 3.